These are the lecture notes for Chapter 4 on cell structure and function. And as you'll see in the next slide, there are 8 sections. I'll try to go through them as quickly as possible. Most of you have heard of the cell organelles before, and a lot of this information you've heard before if you've taken biology at all. And if you haven't, I'll go slowly enough, and you can always stop and rewind, if you need to hear something again. But the first part is that one of the most important theories in biology is called the cell theory, and there are 3 components of the cell theory. So I'm going to read them first. All organisms are composed of cells. And then the second part is cells come only from pre-existing cells. And then the third part is that cells are the basic units of structure and function in organisms. So in the late 1800s, there were German scientists that are given credit for the evidence that led to the cell theory. Matthias Schleiden was a German botanist. That means he studied plants, and he determined after looking at plant tissue under the microscope that all plants are composed of cells. And then Theodor Schwann, a German zoologist who studies animals, he kind of did the same thing. He studied animal tissue under the microscope and determined that all animals are composed of cells. And then the German physician Rudolf Virchow is the one that's given credit for the second part, cells come only from pre-existing cells. In other words, a cell has to divide to make 2 new cells, and that's just how it works. Cells don't come from anything else. They come from other cells. So one original cell had to exist, and then once it came into existence, that cell is what gave rise to all the other cells. Okay, so what we're seeing on this slide number 4, is a cross section of a leaf. In fact, this looks like what do we call those plants? Hibiscus. The ones that have like the big snowball looking flowers. But this leaf, if you tear it in half and look at it in cross section under the microscope, so we're really looking at the edge of the leaf here, you can see the top layer of cells. You can see the layer under that, which are like tall, skinny cells that are lined up parallel to each other. These cells are actually, it's called mesophyll tissue, and these are the cells that contain the chloroplast that are up near the top of the leaf and have access to sunlight. And those are the cells where most of the photosynthesis takes place. And then you have the cells that line the respiratory tract of this rabbit. And actually, this tissue looks much like it would in a human. The cells that line our respiratory tract are lined with cilia. The tissue itself is called pseudostratified ciliated columnar epithelial tissue. And I don't expect you to know that right now. I'm just telling you, for your information. But the top part here that's pink, looks kind of fuzzy. That's because it's lined with cilia. And cilia are little hairs, and what they do is they sweep the mucus up and out of your airways. So anything that's any possible pathogen or toxin that could irritate or cause an infection is trapped in the mucus, and then you cough it up. And it helps to keep you from getting, lung infections. This is a slide that shows you different types of cells and their sizes, as well as sizes of like the largest organism on Earth. Here, the whale, the blue whale, I think it is. But the bottom line is I really don't get into that too much. So the one thing about cell size that I do want to focus on is that we know that cells are small. They're so small that we have to use a microscope to really see them, in any kind of detail. So there's a reason that they are so small, and they are small, and it has to do with their surface area to volume ratio. The outside of the cell, which is composed of the plasma membrane, in animal cells at least, some cells are surrounded by a cell wall outside of the plasma membrane, but the plasma membrane or cell membrane is the structure that really makes up the surface area. So that's the total area of the surface of the cell. And then there is the volume, which is the total amount of space that the cell takes up, on the inside. So cells are small because that gives them a larger surface area to volume ratio. The reason, and this is important, that's why I'm repeating it, the reason cells are small is because that gives them a larger surface area to volume ratio. And the advantage is that it allows them to take in more molecules and get rid of more waste. More surface means more nutrients that can come in and more oxygen that can come into the cell. So, and this is just an example that you can look at on your own if you want to, but it's just showing you a large cell in the shape of a cube so that we can actually calculate some things. And then 64 very small cells that together make up the same volume as this one large cube on the left. So the surface area of the cube on the left is going to be, it says it's a 4 centimeter cube. So it's going to be 4 times 4 because it's length times width, right? So the surface area is 4 times 4 or 16. And then we have to remember there are 6 sides. So 16 times 6. I know this because it actually is the same calculation. I'm going to sound like such a dork, but it's the same calculation I have to do to calculate how many hours that I teach in a semester in a course. We have 6 hours a week in 16 weeks, so we actually have 96 hours total from that. But anyway, that's the only reason I knew that math in my head. But the 96 square centimeters is the surface area of the cube on the left, and the volume is 64. So when you actually work that out, it comes out to be a ratio of 1.5 to 1, 1.5 to 1, okay? So that's the ratio of the surface area to the volume, and the volume is just 4 times 4 times 4, and that comes out to be 64. So 1.5 to 1 is the surface area to volume ratio of this large cube here. If we come over here and do the surface area to volume ratio for the 1 centimeter cube, it's going to be 1 times 1 times 6 is going to give us our surface area. And I'm just going to do it for one of them. So the surface area here would be 6, and that's where that 6 comes from. And the volume would be 1 times 1 times 1, which is 1. So again, math might not be your thing, but you hopefully can see that there is a lot more surface area compared to volume in the small cubes than in the large cube. The surface area to volume ratio is larger in the small cubes. But I always give this example. If you think of these cubes as presents, like Christmas presents, birthday presents, whatever, and think about the surface area as the wrapping paper, which would take more wrapping paper. Wrapping the cube on the left, it's got the same volume inside, but how much wrapping paper would it take as compared to wrapping all 64 cubes on the right, even though their total volume is the same as the big cube on the left? So obviously it would take much more wrapping paper to wrap all 64 of these cubes, and that wrapping paper is the surface area, and that's what we need. We need as much surface area as we can get because that just makes the cell as healthy as it can be. It allows the cell to take in maximum nutrients and get rid of as much waste as it needs to get rid of. Okay, there is also a section in here on microscopes. You're really doing that in lab, so I'm going to skip it just to save time because this is a long chapter. We do use in our lab, I will say this, we use what's called a compound light microscope, and it has an ocular lens that you look through. In fact, the ones we use in our lab have 2 ocular lenses, and you can actually look through both lenses with both eyes at the same time. But there's the ocular lens, which is where your eyes look through, and then there are lenses that have different magnifications called objective lenses. So that's why we call it compound, because there are 2 lenses involved, and then light because you have to have a source of light in order to see the specimen on the slide. But our microscopes can magnify up to 1,000 times if we use the highest objective lens. And then there are electron microscopes, and those obviously, I mean, you can see the difference in the pictures. They give way more detail and magnify thousands of times more than what the light microscope does. But the light microscope is a useful tool in biology. And here you can see some images that were taken through a light microscope of the same thing. These are all cheek cells. And like from the inside of your cheek, you just take a Q-tip, you swab the inside of your cheek, and you put it on a slide. And sometimes, like this one doesn't appear to have stain on it, but but these do. So sometimes you'll add a stain, and sometimes there'll just be different ways of contrast, different types of contrast that you can use. And it'll give you different views of that same cell. It's the very same cell, same type of cell anyway, just using different techniques. So there are a lot of different techniques to help you to see, various different details inside of the cells that you're looking at. Now, moving on, there are 2 types of cells, and you need to learn this and learn it good, because this comes back to you in almost every chapter, and it'll come back in Bio 112. If you've never learned this before, you need to know there are 2 types of cells. There are prokaryotic cells and eukaryotic cells, and the difference between them is mainly this. The prokaryotic cells lack a membrane-bound nucleus. They do not have a nucleus. They do have DNA, but it is not surrounded and enclosed inside of a nucleus, whereas eukaryotic cells do have a nucleus. Okay, so for the organisms that exist, that we know about on this earth, that are composed of prokaryotic cells, they can be placed into 2 different domains. There are 3 domains of living things, but only 2 domains have organisms that are prokaryotic, and that's the bacteria and the archaea. Both of these are, bacteria and archaea, are prokaryotic, and they're also unicellular. In other words, they're only made of single cells. They're not multicellular like human beings are. Okay, and for prokaryotes, they occur in 3 basic shapes. Round or spherical is coccus, or plural is cocci. Rod-shaped or oval is bacillus or bacilli. Spiral can be spirillum or spirochete. And then the cell envelope of a prokaryote, this is the entire structure that surrounds the cell. It's going to be the plasma membrane is the innermost layer, and it is a double layer of phospholipids, or a phospholipid bilayer. Outside the plasma membrane is a cell wall composed of peptidoglycan, and that will be important at some point, so make sure you know that prokaryotic cell walls, and actually we should be clear, peptidoglycan is only found in the cell walls of bacteria, so not archaea. And then we have the glycocalyx, which is outside the cell wall, and it can either be a capsule or kind of a slimy layer. And you can see the shapes here, the coccus, the spirochete, the spirillum, and the bacillus shapes.